Okay, well, thank you, Rachel. We'll go ahead and get started then. Um, para las personas que desean interpretación al español, sigue al fondo de la pantalla y se encuentra el enlace para la interpretación y Diego Cheles está interpretando para nosotros hoy. So, bienvenidos a todos ustedes. So, welcome everybody to this is the first of a three-part online series on practical training on nitrogen management in organic production of vegetables and strawberries. And we're very happy to have all of you. We have a good group of people that have signed up. And we're very fortunate today. We have in our first session, we're going to go through some practical aspects of nitrogen management and understanding of nitrogen management. We have some, we're very fortunate for the speakers we have today. Daniel Geisseler is a extension specialist at UC Davis, specializing in nutrient management. And Margaret Lloyd is a farm advisor in Yolo County, doing a lot of work with nitrogen management and especially with organic. And then some of you may remember Louise Jackson, worked here in the Salinas Valley for many years. And she is just understands better than anybody that I know the role of microbes in transforming nitrogen and making it available for plant growth. And she's going to be our third speaker today. So I think without any further ado, we're just going to launch into our first speaker. It'll be Daniel Geisseler. And if you have questions at the end, put them in the Q&A. And that's how we're going to generate the discussion. So please don't be shy, ask your questions, and we'll try and get to all of them. So thank you. So Daniel.
Great. Thank you very much, Richard, for the introduction, for organizing this webinar, and thanks everybody for joining. So my topic, I will cover two topics. First, I will briefly talk about nitrogen in the soil and the factors that affect nitrogen mineralization. And then in the second part of my presentation, I will talk about nitrogen mineralization from soil organic matter, present some results from our own research. There are different pools of nitrogen in the soil. Most of the nitrogen is in the form of organic material. That's probably more than 90%. That's soil organic matter, but also when you incorporate cover crops or organic amendment, most of it is in the form of organic nitrogen. On the other hand, plants take up most of the nitrogen either as ammonium or most of it in our agricultural systems as nitrate. So these are the two forms that compose most of the nitrogen taken up by plants. Organic nitrogen can be converted to mineral nitrogen by soil microorganisms, and that's what we call nitrogen mineralization. So whenever you add a residue or an organic amendment, it first needs to be converted by soil microorganisms to ammonium, and other microorganisms may convert it to nitrate, and then these are the two forms plants can take up. So I'd like to focus briefly on the factors that affect nitrogen mineralization, just in general, independent of the form of the organic material that's added or that's in the soil. The main factors are soil temperature, soil moisture, then the quality of the organic source, and also management of the crops or of these or of organic amendments. Let's start with soil temperature. Microorganisms become more active when temperature increases, at least within the range of soil temperatures we observe in our agricultural ecosystems. So microbial activity, which includes decomposition and nitrogen mineralization, is very low at low temperatures. And that's why we keep our food in the fridge, because it doesn't spoil. Microbes cannot spoil it, or much slower when temperatures are low. And temperatures increase, microbial activity, including nitrogen mineralization, increases more exponentially, not just linearly. So that means in summer, microorganisms are much more active than during the winter. Here's just an example. Most CIMI stations, they measure soil temperature at 6 inches. And as we all know, during the winter, soil temperature is low. Here, the brown curve is for the Salinas weather station, the black curve for Davis. It starts in January all the way throughout the year to next January. In Salinas, during the winter or in early January, soil temperature may drop all the way to 50 degrees Fahrenheit, and then slowly increase, reach a maximum in July, August with some 70, 75 degrees, and then slowly decrease again. Soil temperature, of course, has a similar temperature curve over the year as air temperature. It's just a little bit behind, because it always takes a while to warm it up or for the soil to cool. In the Central Valley, the temperatures drop a bit more during the winter and increase a bit more during the summer. But the curve is very similar, and due to the changes in air temperature. Soil moisture is also very important. Here I show an example for a silt loam. On the horizontal axis, we have the volumetric moisture content, and the area I shaded here is between field capacity to the right and the permanent wilting point to the left. So that these are is the water content important for plants. And the vertical axis, that's just a relative measure. When nitrogen mineralization is optimal, which is at field capacity, that factor is 1. And then if it's slower, the factor decreases. So we can see microorganisms as plants are happiest when the soil is moist, when it's near field capacity. When the soil dries out, microorganisms are less active. At the permanent wilting point, which is the moisture content where plants start wilting when it's that dry because they cannot access water anymore, microorganisms can still be active, much less than at field capacity, but there are still small pores that are filled with water that may be too small for plants to access the water, but microorganisms are so small that they can still be active in these pores. But when soil moisture drops even below the permanent wilting point, then it becomes too dry for most soil microorganisms, and nitrogen mineralization is very low or even stops when the soil is very dry. So when we combine these two curves, and I did these calculations for a cropping system in the Central Valley, but it would look very similar for the Central Coast. So when we look at the soil temperature during the year, combine it with the temperature response, and look at soil moisture during a year and combine it with this moisture curve, we get something like this. And I'll show you this graph here a bit bigger on the next slide. So what that shows us, again, from January all the way to January, and a relative factor, is that during the winter, nitrogen mineralization is low. And that's mainly because temperature is low. So it's temperature limited, even though when we have rainfall during the winter, soil moisture content would be optimal, but temperature is just too low for microorganisms to be very active. But then in spring, when temperature increases, microbial activity increases too. And it's highest during the summer, because that's ideal. The soils are moist and they are irrigated and they are warm. So ideal conditions for microorganisms. Then in fall, microbial activity drops again. This curve, I created this curve for a processing tomato system where the tomato was harvested in August. On the Central Coast, where you may grow 2 crops or a crop that is harvested much later, you wouldn't see that early drop in microbial activity. So this drop is due to the harvest of the tomatoes and then no further irrigation. If you have a longer season crop or you plant another crop and you continue irrigating, then this curve would remain relatively high for another month or 2, but then finally drop again because the temperature in fall becomes cooler, so microorganisms are more limited again. Then another factor that plays a big role in how much nitrogen is mineralized from an organic source is the quality of that organic material. And one quality measure that is very important is the nitrogen content or the carbon to nitrogen ratio. Both of them are closely related. This is a graph just showing how the nitrogen content of a residue in this case, but it would be very similar if we look at organic amendments, how that affects nitrogen mineralization. This is data from laboratory incubations over a period of 12 weeks. And the vertical axis is in % of the total nitrogen added with that residue. And what we can see when we have a residue with 3 to 5% nitrogen, that could be broccoli residue or lettuce residue, could also be a legume cover crop. We see that nitrogen mineralization takes place very quickly. So a lot of nitrogen is released and becomes plant available immediately. And then the curve flattens off, but by the end of the 12 weeks here, up to 60% of the total nitrogen is released and has become plant available. And the rest, the 40 or 50%, that would have been incorporated into microbial biomass. It may be released later, or it may be turned into soil organic matter. When we have a material with a lower nitrogen content, that could be, for example, tomato residues or melon residues with a nitrogen content of 2 to 3%, also a young cereal cover crop. What we see is that we may still see nitrogen mineralization, but it's a bit slower, and the total amount of nitrogen mineralized over the same period of time will be lower. And more of the nitrogen will be either released later or incorporated into soil organic matter. When we have a material with a very low nitrogen content, probably less than 2% in the case of residues, that could be some straw from cereals or corn stalks, then we may even see some nitrogen immobilization, which means that the amount of nitrogen that's plant available may initially actually decrease because microorganisms take it up. And only later, after a while, we see nitrogen mineralization or an increase again in the amount of plant available nitrogen. So why is that? Why do microorganisms sometimes mineralize nitrogen, sometimes immobilize nitrogen? The goal of the microorganisms in soil is to decompose the residue or the organic material. And they need nitrogen and carbon as building blocks for their own biomass. So if they want to grow or multiply, they need carbon, nitrogen, and other nutrients as well. Carbon, just as we do with our food, they also need organic carbon molecules as an energy source to respire and to gain energy. Now, if the ratio between nitrogen and carbon is narrow, there will be excess nitrogen. They don't need all that nitrogen for their own biomass. So in that case, the microorganisms may release some of that nitrogen as ammonium into soil solution. basically at that moment, the ammonium is a waste product they can't use. And that's what we call nitrogen mineralization. On the other hand, we may have some residue or organic material with a lot of carbon, but hardly any nitrogen. So the microorganisms have all this carbon they could use to grow and to multiply, but not enough nitrogen for their own biomass. So if they find some available nitrogen somewhere else, for example, in soil solution, if there is nitrate or ammonium in soil solution, they may take it up and incorporate it into their own biomass. And that's what we call nitrogen immobilization. So the nitrogen is not lost, it's still there in the soil, but it's tied up in the microbial biomass and at the moment not available to the plants. It may become available later at one point when the microorganisms die and decompose themselves. Then some of that nitrogen will become plant available again. But at the moment, it's no longer plant available. The threshold we often use is that we say when the carbon to nitrogen ratio is below 20, we normally see net mineralization or an increase in plant available nitrogen. When the carbon to nitrogen ratio is higher than 30, we can expect net immobilization. These are not fixed thresholds, and you will see next time with organic amendments, we actually found that it may be, the threshold may be a bit lower than 20, but you can, just as a rule of thumb, it's if the CN ratio is less than 20, you can expect some nitrogen mineralization. If it's higher than 30, it's very likely that the material is going to immobilize nitrogen and make, in the short term, at least nitrogen less available in the soil. Then, in addition to the carbon and nitrogen ratio, the availability of the carbon and nitrogen also plays an important role. And I'd like to illustrate that here with a graph where we see the nitrogen mineralization in pounds per acre over a period of 100 days. If broccoli residue is incorporated, and broccoli residue has roughly a C/N ratio of 10, and it may contain about 210 pounds of nitrogen. If that's incorporated, we see a very quick release of the nitrogen. Up to 130 or up to 140 pounds of the nitrogen in that broccoli residue is released, and most of it within a month or so. On the other hand, when we look at soil organic matter, which has a similar CN ratio, very often soil organic matter has a CN ratio of 10. If you think about a soil with 2% in the top foot, it may have 3,500 pounds of nitrogen per acre, or up to 2 tons. So there's much more nitrogen in the soil organic matter than what we added with the broccoli. But this curve shows the nitrogen mineralized from the soil organic matter. So even though we have much more, soil organic matter decomposes very slowly compared to broccoli or other plant residues. And you see by the end of the 100 days, probably some 20 pounds or so per acre may have been mineralized, just as an example. So the quality of the organic material plays an important role. Soil organic matter is a very complicated molecule. It's much harder to degrade than plant residues. It may also be protected by some clay minerals or physically protected in soil aggregate. So that these are all reasons why soil organic matter decomposition is much slower than the decomposition of residues, and therefore nitrogen mineralization is much lower. So that the quality always plays an important role, not just the CN ratio. Then the last point that's important to take into account is the management of the residue or the organic amendments. Here's a slide from Richard Smith. He did a very interesting study where he used pelleted organic fertilizer, a 442, and either left it on the soil surface, this is the red curve, or incorporated into the soil. The vertical axis is again the percent of the total nitrogen in that material released, and it was over a period of 70 days. And what you can see is when a pelleted material is left on the soil surface, the nitrogen is released much slower. And that's probably due because if something is left on the soil surface, it dries out during the day. And so microbial activity is almost stopped or slowed down because it dries out. On the other hand, when it's left in the soil, it may remain constantly warm. It's in contact with soil minerals or with microorganisms in the soil and decomposes much faster. So it makes a big difference when you use the same material, whether you leave it on the soil surface or whether you incorporate it. Then briefly in the second part of my presentation, I would like to touch on nitrogen mineralization from soil organic matter. As I've already mentioned, there's a lot of nitrogen in the soil. If you have soil organic matter content of 2%, there may be up to 2 tons of nitrogen just in the top foot of the soil. So that's much more than our crops need. But we all know if we don't fertilize, our crops will likely be nitrogen deficient. That's because organic matter is not that easily degraded by soil microorganisms. But still, soil organic matter, mineralization from soil organic matter can make a nice contribution to the nitrogen used or needed by the crop. So it's beneficial to know how much can we actually expect to be mineralized. And that was a question we had in one of our studies where we went out in spring before the season started and collected undisturbed soil cores from the top 6 inches of the soil. And we incubated them at an optimal moisture content at different temperatures for 10 weeks. And after 10 weeks, we measured how much nitrogen and ammonium was in these cores. And we also measured it at the beginning. So we saw the increase, or we could calculate the increase in plant available nitrogen. We used 3 different temperatures, and based on these results, we could create the temperature response curve I showed you earlier. But I will focus now on the incubation at 77 degrees Fahrenheit. Our samples were taken, most of them in the Central Valley, some of them in Salinas. We also sampled some soil in the Delta with a high soil organic matter content, and in Tule Lake, where the soil also had a higher soil organic matter content than we are used to in the Central Valley and Salinas Valley. And this is the nitrogen mineralized during the 10 weeks. On the y-axis is the amount, and each bar represents 1 soil sample. And you can see very easily the soils in the Delta and Tule Lake that have a higher soil organic matter content, they mineralize much more nitrogen. And nitrogen mineralization was lower in the Central Valley and Salinas Valley. The soils here in the Salinas Valley had relatively low nitrogen mineralization rates, although I wouldn't generalize that. I think that was just the sites we sampled. So I wouldn't conclude based on this data that nitrogen mineralization is lower in the Salinas Valley. And I'll come back to that in a moment. So we measured a lot of soil properties to see how can we relate soil properties to the amount of nitrogen mineralized. And we did it separately for the high organic matter content soils and those with a relatively low soil organic matter content. And I'll focus now on these soils. And what we found is that the best way for these soils to predict how much nitrogen is mineralized is to use total carbon which is a measure for the soil organic matter content, because roughly half of the soil organic matter is carbon. And FDA hydrolysis, that's a measure of the microbial activity in the soil. And we could probably say here it's also a measure of the quality of the soil organic matter. And then silt as a measure for texture. And you can see on the horizontal axis is what we measured in our incubation, and on the vertical axis, what our model predicted. And if our prediction were perfect, all the symbols would be on this diagonal line. And you can see our prediction isn't bad. We definitely capture most of the variability, but still there are some soils that are a bit far away. So where other factors other than the three properties or the properties we included in our model play a role. So we're still working on that. We are still trying to get a better estimate of how much nitrogen is mineralized so that we could actually make site-specific estimates. At the moment, we're not too confident yet in site-specific estimates of nitrogen mineralization. But we also used another approach because lab incubations are a great tool to compare different soils, but they are done under constant temperature, constant moisture. So that's quite different from the field. So we were also wondering, how much can we actually expect? How much nitrogen mineralization can we expect in the field? And we did that for the Central Valley soils, where we looked at what is the input in organic nitrogen. So not mineral fertilizer, just organic forms of nitrogen. And the soils were all collected on conventional farms. So that means these soils had no history of organic amendments or legume cover crops. So in these cases, if we grow a crop every year and it's fertilized with mineral fertilizers, then the input of organic forms of nitrogen is with plant residues. So the above ground residues left in the field. The roots, they decompose at the end of the season. So that's an input in organic N. We also, during the growing season, there may be some root exudates that are released, and some of them contain organic forms of nitrogen. And if we see a decrease in soil organic matter, that would be another source of organic nitrogen added. So we did some budget calculations, how much is actually the input. And our hypothesis was that the input needs to equal output in the long term, especially when we take changes in soil organic matter content into account. And output is really output of organic forms of nitrogen in the system. That's really mainly nitrogen mineralization. So what we found is for the crop mix we had at the site, that on average about 70 pounds per acre are returned in the form of residue, 70 pounds of nitrogen per acre. Some 23 pounds in the form of rhizodeposition, or the 70 pound, by the way, that includes the roots. Sorry, I forgot. And there may be some decrease in soil organic matter content. We estimated that at 70 pounds per acre. So that means the total organic input is somewhere between 70 and 110 pounds per acre over a 12-month period. So that's our estimate for Central Valley soils. On the Central Coast, these numbers may be slightly higher because growers often grow 2 vegetables a year, although I don't think they would be double that, because the residues incorporated in the Central Valley, let's say in a lettuce broccoli rotation, a lot of the nitrogen is released immediately and isn't converted into soil organic matter. So on the Central Coast, if we look at the nitrogen mineralization per day, then we will probably see from soil organic matter, it increases. But then, let's say if we have a lettuce crop and we incorporate the residues, a lot of it will be immediately mineralized or over the next few weeks, and the same with broccoli. So with these residues from the vegetables on the Central Coast, a lot will be released immediately, and a smaller proportion will be incorporated into soil organic matter. But still, these numbers may be a bit higher, so the total nitrogen mineralization based on the budget may be a bit higher. But back to the Central Valley, I've already shown you this graph here, where we look at the nitrogen mineralization during the season. If we take a crop that is grown from May to the end of August, for example, a processing tomato, during this period of time, roughly, it's only 4 months, but because it's during the summer, roughly half of that nitrogen is mineralized from soil organic matter. So somewhere between 35 and 55 pounds per acre during a 4-month growing season. And this estimate is pretty close to estimates from other areas where they looked at nitrogen mineralization with other methods. So at the moment for the Central Valley, that's our best estimate. On the Central Coast, depending on the crop rotation, it may be probably a bit closer to the 110 pounds per acre. For organic systems, we created a worksheet where growers can calculate a budget, and you will get a link to that worksheet. There we simplified it a bit, and you can look up the values for individual months, whether you're on the Central Coast, in the Sacramento Valley, or in the Imperial Valley, and you can choose a soil organic matter content that's closest to what you have in the field. So these are rough estimates, but you can also see the numbers per month are relatively small. So if you're off by 10, 20%, it's not that much of a mistake, or it's not that much off. But at the moment, these are our best estimates, and we hope to get some better data in the future. So that's my presentation, a brief introduction to nitrogen in the soil and soil organic matter. So we have some time for questions.
Okay, Daniel, thank you very much. We have one question in the question and answer from Laura with the RCD in Monterey County. She says, can you share a list of labs available that are available to the general public to provide the FDA hydrolysis analysis for soil samples?
That's a good question. And as far as I know, at the moment, commercial labs don't offer that yet. So it's not a difficult method. It could be easily done, but it's not a method that's offered. It's sometimes used for soil, it's often related to soil health measures. So there's a possibility that probably some labs that will focus on providing soil health estimates for growers will offer it in the future. But at the moment, that's not offered yet, as far as I know, by commercial labs.
Those measurements that you just mentioned that the labs provide, what are they measuring?
For soil health?
Yeah.
Or,
yeah.
I think in California, we don't have, not aware of labs that measure soil health. Cornell does some soil health measurements, and they measure POC, permanganate oxidizable carbon, or a short incubation where they measure CO2 release to get at the question of the quality of soil organic matter, of the quality of the soil organic matter. These are probably the most common measures at the moment. Some of them may offer protein content and different measures, so it's not standardized yet.
What do you think of those measures? Do you think they're valuable?
Well, they all have their strengths and weaknesses. I think we probably need a mixture of, some of them are better related to the the short-term availability. Others are better related to the amount of organic matter in the soil. So I think a combination is best, but I also think we really need regional data. We probably shouldn't just use an estimate of soil health that was developed in the northeast of the US, because our conditions are different.
Okay, all right, well, thank you. We have a few more questions here for you. Do you make the mineralization worksheet for the organic Central Valley farms based on soil organic matter? So I guess you're referring to the worksheet. Is that based on soil organic matter?
So yeah, our estimates of nitrogen mineralization from soil organic matter in the worksheet is based only on soil organic matter content. So we didn't include other factors, but then we use the temperature curve to calculate how much may be mineralized every month. But because I've shown you, we don't have that good data yet for site-specific estimates, so we used a conservative approach and based these estimates in the table I've shown solely based on soil organic matter content, or yes, soil organic matter content and temperature in a specific month.
Okay, all right, and then here's a question in Spanish. What pH is the mineralization of nitrogen optimized, or how sensitive is that to pH?
It's not that sensitive. It's definitely best around a neutral pH, but we probably wouldn't see big differences, let's say between or within a pH of 6 to 8. When the soils become very acidic or very alkaline, then we would certainly see a drop off or a lower nitrogen mineralization rate. But it's less sensitive than probably other processes in the soil, because a lot of microorganisms that are adapted to very different conditions decompose organic matter or organic material. So it's a wide variety of microorganisms that can decompose and may release nitrogen into the soil. So it's not that sensitive to pH, but of course, if it gets very acidic or alkaline, then it will drop off.
Okay, I mean, just looking at the soils in this area, that range from 6 to 8, most soils fall in that range. Not all, but most do.
I think so. I wouldn't expect in our areas the pH to be really limiting factor.
Yeah, so it's not a much of a factor. Okay, here's another question. Would leaving a vegetable crop residue like broccoli on the soil surface in the fall reduce the chance of nitrate leaching from the residue in the winter?
So it would decompose much slower. So yeah, it depends on when it is incorporated. I mean, decomposition doesn't stop, it will decompose. So it depends a bit on how much time it has to decompose before it gets really cold, but it will definitely decompose slower. So there will be, or the nitrate will accumulate much slower and potentially reduce the risk of leaching of nitrate, yes.
Yeah, it might be kind of similar to that fertilizer example that you showed.
I would expect it to be very similar. It's not zero, but it's slowed down.
Yeah. Okay, and then I guess we have time for one more question. But people, if you have more questions, you can send them through the question and answer to Daniel, and then Daniel, you could type them in, but we'll go with this one last one. Is there a good method for determining the quality of nitrogen source from organic amendments that have the same C to N ratio?
Patricia will cover some of that in the next next meeting, and there are definitely differences. It's a bit hard to generalize. One way, probably one generalization is that if the material is composted then nitrogen availability will be a bit slower than with a fresh material. So if you have a fresh manure, it will become available a bit faster than when it's a compost. But other than that, it's kind of hard to point to other properties we generally know about the organic material. So the CN ratio is definitely the best way to look at to get some good idea of how much is available.
Mm-hmm. Okay. Well, we'll leave it there for now. For Daniel, thank you very much for the presentation and for answering those questions.
Thank
We'll
you.
move on to our next speaker. But like I said, if you have more questions for Daniel, just go ahead and put them in the Q&A, and then Daniel, you can respond typing them in. Okay. Thank you very much. All right, now we'll move on to our next speaker. Is, as I mentioned, Margaret is a farm advisor up in Yolo County, and she's been doing a lot of fantastic research on cover crops and soil organic matter and nitrogen made available for plant growth. And so Margaret today is going to talk to us about nitrogen made available from cover crops. So Margaret.
Great, thank you, Richard. Okay. How does that look?
Looks great.
Okay, thank you. Thank you, Richard, Joji, and everyone for putting this workshop together. It's really a pleasure to be here with all of you. I wish we could all be in person, but we'll have to save that type of fun for another workshop, another year. As Richard said, I'm the Small Farms Advisor for Yolo, Solano, and Sacramento counties, and I have the unique privilege to have a program assignment focused on organic production. So today I'll be talking about cover crops and crop residue in the nitrogen budget. And my goal is to help build your ability to understand and estimate the contributions of crop residue to the nitrogen budget. So whether this is for nitrogen planning, reporting, decision-making when selecting a cover crop, or terminating or timing of activities, I hope this information can provide you with some new and useful ways to navigate those scenarios. This here is a conceptual figure. Oh, wait, can you see it, or is it blank?
We can see it.
Okay, sorry. This here is a conceptual figure that we designed using data from multiple studies from the Geisseler lab to illustrate the relative contributions of the multiple nitrogen sources in a summer growing scenario in Yolo County. And as organic farmers, the contributions from many sources is an important one to really grasp and think about how much nitrogen is coming from all of them, because it's really a collective contribution. And sometimes it can be framed that we think more about our amendments as being our nitrogen sources, when really it's many actions accumulating over time. So on the bottom here, we have the soil and the soil organic matter. So a lot about what Daniel just introduced. In this light green band, we have cover crops or crop residue. In this blue band, we have, this represents a compost application of poultry manure. This purple band is a granular fertilizer at 4%. And on top, we have this orange band, which represents some nitrogen coming from the irrigation water. And this red dashed line is just to represent a tomato, a fresh market type tomato, an uptake curve for how much nitrogen it's using over the season. So I have this here just to give the idea of all of these contributions from many places are really building the nitrogen program in an organic system. Now on the coast, the ratio of contributions is likely similar, but with the coastal climate, the summer peak may be less pronounced. And so that nitrogen release and uptake is likely more spread out across more months. So the amount of nitrogen made available from crop residues largely depends on the biomass of the residue. So how much material is out there, the nitrogen content of those residues, and this C to N ratio and total nitrogen content, which is a lot of what Daniel just introduced. And then these factors that are driving and influencing the microbial activity, soil moisture, and whether that material was incorporated or left on the surface. So I'll be going through each of these and discussing how the quantity quality affects the availability. So first I'll talk about the biomass of the residues. And the vast majority of nitrogen in cover crops is in that biomass, that above ground biomass. If you're ever asked how much nitrogen or nitrate is coming from your cover crop, at a minimum, you'll need to know two pieces of information, and that's the biomass and the tissue and content. So the first step is getting an idea of this biomass and what drives the production of the biomass. Can you look at this picture and get an idea of how much biomass is out there? This picture is just under 3 tons per acre dry weight of cover crop residue. Here's another image. So we have in the foreground or in the middle here, a vetch cover crop. This represents 2 tons per acre of biomass. So when during the season is biomass accumulating? Here's a figure showing the growth of a cereal crop. So some cover crops are cereal, some are legumes. So in the case of cereal crops, after the boot stage, so right about here, the rye cover crops can double in biomass with little additional N. So they're really putting on the bulk of the biomass in the latter half of their growth stage. So shortly after boot stage, most of the N movement is in the translocation, and the rate of N uptake decreases significantly. So this curve shows the biomass accumulation over time. And so what you can see is right about here, we just passed boot stage, and we see this rapid biomass accumulation, this buildup of biomass. So when thinking about getting more nitrogen into our fields, we're doing that more quickly after this boot stage. Now, to achieve this growth, cereal crops, the non-legumes, scavenge for residual nitrogen in the soil. The grasses, in particular, have these deep, efficient root systems, and they scavenge for residual nitrogen in the soil profile. And they have good potential to capture plant-available nitrate that is in the soil, preventing it from leaching into subsurface groundwater or running off with surface water. So that's one of the advantages. However, they will be limited by the available N in the soil. So these two lines represent a scenario in which there was sufficient nitrogen in the soil, high amounts of residual soil nitrogen, nitrate, and a scenario where there were lower nitrate levels. And so you can see how that influences the potential for the biomass accumulation. In the document that Daniel was talking about, which we will share in the link, we have a lot of the tables that we'll be talking about throughout this workshop. So this is one of the tables in which we have listed out a sort of some baseline common estimates for the amount of cover crop biomass from some common cereal, legume, brassica cover crops. Just to at least give that baseline idea. But refining the biomass production on your farm has to come with some amount of sampling and training your eye to be able to gauge what 2 tons, 3 tons, 4 tons of biomass looks like. But you can see here how if we just look at oats and rye, they're the highest biomass producers, whereas the vetch only type species are the lowest. And the mixes with some cereal, legumes, bell beans, mustards are in this intermediate range. Now thinking about legumes. So the vast majority of N in the cover crop is in the above ground biomass. And this is regardless of whether the nitrogen is coming from residual soil nitrate in the soil, or if it's fixed from the atmosphere by the bacteria in legume roots. So legume, as many of you know, the legumes differ from grasses and other non-legumes in their ability to form symbiotic associations with root nodulating bacteria called rhizobia. And this symbiotic relationship provides, the plants are providing the root nodulating rhizobia bacteria with the carbohydrates, and in exchange, the rhizobia are providing nitrogen. And this exchange is not free. The legumes can invest as much as 20% of their net photosynthate production to supporting those rhizobia. And the rhizobia share nitrogen that they could otherwise be using for self-growth and reproduction. And this exchange occurs in those roots. However, that nitrogen is translocated to the above ground plant growth for the most part. And to give you an example, in a hairy vetch and Austrian pea cover crop that was 100 pounds of nitrogen per acre in the above ground biomass, the roots had about 10 pounds of nitrogen per acre. And when you also look at the makeup of those roots, they had a tissue content of less than 2% nitrogen and a C to N ratio of more than 20:1. So the plant available nitrogen, the PAN, that release was close to 0. So in that example of a hairy vetch Austrian pea cover crop of 100 pounds of nitrogen per acre, that nitrogen was coming from the above ground biomass and not from the roots that are left in the ground. So legumes, this ability to take the atmospheric N means that they're providing a net input of N to the soil when they're incorporated. One feature of a legume cover crop is that they typically have higher N content in their tissue. And they will usually accumulate N longer in the spring than non-legume cover crops. So thinking about the timing and which scenario best fits into your planting windows, and whether an earlier accumulating, a crop that accumulates biomass earlier in the season or later in the season, these can be selected based on the crop species. So now thinking about when is nitrogen highest? So we just spoke about biomass accumulation. Now thinking about nitrogen. So I discussed how after boot stage is when that biomass really accumulates. However, it's at boot stage that the leaf tissue N content is highest. So when we see what that curve looks like, this curve is showing the percent nitrogen. And so we see it highest earlier in the season, right before boot stage, and then it starts to decline. So that's the other piece of information, is understanding this tissue N content. And building on the table we looked at earlier, we can see our top 2 candidates here as a cereal cover crop, that the tissue N content is less than 2%. And as we scroll down, these vetches are among the highest at 3.5, 4.5 or so % tissue N content. And again, these mustard, bell beans, and mixes are intermediate. So you can see the sort of decision and challenge here in choosing biomass and tissue N content. Because so then when we think about termination, we think about how do we maximize biomass and maximize tissue N content. And so identifying that point in the season can help with some termination decisions if there's flexibility in that decision moment. So here we can see the biomass accumulating, the nitrogen content decreasing, and terminating somewhere in here is going to maximize our total N. So that's going to be the tissue content times the biomass. The legume cereal cover crop mixes are one way to sort of capture the qualities of both a cereal crop and a legume crop. They can provide this robust system with the capacity to both scavenge the residual soil N, but also utilize atmospheric N and grow abundantly even in low N conditions. And then this can often lower a total, the tissue N content can be higher when legumes are mixed into a cereal crop. So when we look at the N content of residues, a couple of things are influencing that. It tends to be the cover crop age. So the younger the cover crop, the higher the N content of that species, and the higher the N availability following incorporation. So they're going to tend to also have a lower C to N ratio, be more available, and so that's why they'll be available earlier than older materials with a higher tissue and higher C to N ratio. In general, the legumes and mustards have that higher N content in their tissues, more than 2%, and that's what will allow for this rapid mineralization. Now, cereals can have higher N content, but that's going to be earlier in their growth period when it will be more than 2%. So that's when they're juvenile, and they will have less biomass. So that's the trade-off. So now we've talked about the biomass, the tissue N content, but only a portion of that total N becomes available to the following crop. And so how much becomes available? So the C to N ratio is the mass of the carbon relative to the mass of nitrogen in a substance. And the soil microorganisms have a C to N ratio near 8:1. They must acquire enough carbon and nitrogen from the environment in which they live to maintain the ratio of carbon and nitrogen in their bodies. And because soil microorganisms burn carbon as a source of energy, not all of the carbon that a microorganism consumes remains in its body. A certain amount is lost to CO2 during respiration. And so to acquire the carbon and nitrogen a soil microorganism needs to stay alive, it needs a diet with a C to N ratio near 24:1. So that's where we often talk about these numbers above and below 24:1, as Daniel pointed out earlier. And so when carbon contributions are high, these decomposer microbes will scavenge any free nitrogen in the soil or immobilize it in their bodies as they chew through high carbon materials. Vegetable crop residues typically have a C to N ratio below 15:1, which allows for rapid nitrogen mineralization to begin immediately following incorporation into moist soils. Whereas residues with a ratio of 15:20, as is common with cover crops, those are going to mineralize a little bit more slowly. And then those above 20:1 will be very slow or start to tie up and immobilize some of the nitrogen. And we'll look at that a little bit more closely in a minute. So here are some figures of cover crops and their carbon to nitrogen ratio. So in this first field, this is a wheat crop. The second field is a vetch crop and a veg crop. This fourth field is a mix, and we have a few mixes here. Mix field 4, 5, and 6, and 7. And just to give you a sense of what the range, so here we have a range of 12:1 to 18:1 of the cover crop carbon-to-nitrogen ratio. For contrast, here you can see a very mature cover crop. It has a lot more biomass, but you can see the seed heads and the flowers, and that cover crop is measuring 32:1. And here's the tissue content of those same fields. So you can see this higher C to N ratio has a lower N tissue content, 2.7%. Whereas when we go over here to the 12:1 C to N ratio, there's 3.9% tissue content of nitrogen. So you can see how those two things are related. And then when we revisit the figure that Daniel introduced, we can look at what percent of that tissue might be mineralized based on tissue content. And we'll go through this exercise in a minute. So we look at the tissue N content, the total N in the biomass, and then visiting this figure, I'm going to estimate what that mineralization rate might be. So let's walk through this first one together. I have 1.7%, so that's going to be in this less than 2% band. And so if I draw this line over, I said that it had an estimated mineralization rate of 10%. And then let's do this last one at 4.7% tissue N content. That's going to be up here somewhere. Draw this line over, and I put in it had a 55% mineralization rate. So then to estimate what amount of plant available nitrogen of all the total nitrogen in that cover crop, you'll multiply the biomass times the mineralization rate to get available N. So in this Cayuse oats example, we might expect 13 pounds of nitrogen per acre after that cover crop is incorporated. Whereas in this Lana woolly pod vetch example, we might expect 100 pounds of nitrogen post-incorporation. So in that way, you can get your own numbers and do these types of calculations paired with a figure like this to estimate mineralization. Cover crop residue is going to function similarly to cover crops. And so we can talk about them in the same way and think about them in the same way. Similarly, we want to have that information of C to N ratio or tissue N content. And most of the tissue N content of vegetable residue is going to be 2.5 to 5%. So it's going to be somewhat similar to a leguminous cover crop. And then if you have idea of how much residue you have in the field, you can do that calculation. We have included in that document we've been referencing a table with some estimates of residue. So if we take broccoli, which tends to can have tremendous amounts of residue, especially on the coast where that is prime broccoli growing conditions. And so this is an example yield of 10 tons per acre, and how much N content is in that tissue, or 68% of the total N that was put into that crop still remains in that residue. That's a lot of leftover nitrogen. So then the expected amount of nitrogen in that residue is going to be 240 pounds. So that's the total nitrogen in all that residue. At that point, you would want to do that calculation that we just did here and walk through 240 pounds times a mineralization rate that you choose based on your estimate. And so maybe 3% nitrogen in a broccoli residue. And you can get what a actual plant available nitrogen release might be from that residue. I have this picture here to illustrate the concept that only a portion of all this material that we put in is going to be used by the subsequent crop. So this was work by Louise Jackson, who is here today, who will be talking next. And this is some work in which she tracked how much of the nitrogen from a cover crop was taken up by a subsequent lettuce crop. And what you can see here is that this is just the first planting of lettuce. There were 2 plantings. But in this first planting, combining the shoots and the roots, just over 20% of that lettuce's total N came directly from the cover crop. And where did the rest of that cover crop nitrogen go? A lot of it went into soil organic nitrogen, which is what Daniel was talking about. A small amount remains tied up in the microbial biomass. This section here describes what would have been potentially leached. So I just have this here to further demonstrate or illustrate this idea that our crops are only taking up some amount of the nitrogen from these cover crops or crop residues following incorporation. And a lot of it is being built into the soil. And that's where we get this tremendous ongoing background release of N in organic systems, where we're repeatedly cover cropping, putting crop residues in, adding materials like compost. We build this robust background nitrogen source in our soil. And just to reiterate, what's going to influence how much of that nitrogen we actually get? So in addition to the basic factors of biomass, tissue content, C to N ratio, we have to consider the microbial conditions. So soil moisture is essential for them to do their work. And so that's going to be a driver of what we actually get to see from the microbes. And whether those residues are left on the surface or incorporated. And Daniel touched on this already. But basically, the surface applied residues will decay more slowly than the incorporated residues. So that can be ideal. That can be maybe a strategy that was mentioned before. This idea of maybe leaving broccoli on the surface to delay leaching, or it can be incorporated into a system. So lastly, I encourage folks to try to get some of these numbers for yourself. And I think that doing this exercise of collecting biomass and measuring the C to N ratio and the nitrogen content of that biomass is an important way to begin training your eye to understanding how much is going back into the system and what practices are you doing that are influencing the quality and quantity of that material. And it's quite simple and cheap to do that. So quickly here, I'll give you a demonstration of that. Basically, we have this meter square, which is just a set of PVC pipes measured out to 3 feet by 3 feet or meter by meter, whichever you prefer, but a set area. And you take all that cover crop, you cut it down, take all that material or crop residue of vegetable residue, and you dry it. And then a subsample of that is sent to a lab to get those, the tissue N content and the C to N ratio, and those tend to be under $20, $11, $12, $13, $15. So it's quite cheap. And in this way, you can get real numbers for your field. And after this is done a few times, I believe that you'll be able to train your eye to begin to read what might be going on out there. So you'll be able to plug in your own numbers for tissue N content and cover crop biomass. And then you can use the information that we have provided to get your estimated plant available nitrogen. So in summary, the amount of cover crop N that is made available for vegetable crop growth varies widely, and estimates range from 0 to about 50%. So the unmineralized N from that crop residue and the cover crops contribute to the total N in that soil organic matter and the long-term soil fertility, but is less available in the short term. So some food for thought. The cover crop mixes with a higher proportion of legumes, particularly when terminated before flowering, will release more of their N than later terminated crops and grass-heavy mixes. So I apologize, I didn't quite emphasize that in the legume growth, I talked more about the cereal growth, but in the legume growth, the tissue N content is maximized just before flowering or early flower. So that's where that curve will start to decrease for the legumes. An early spring kill of cereal grains reduces accumulation of carbon more than nitrogen. So the rate of release of the cover crop N into the following crops is faster. In a cover crop mix, low residual soil N will limit cereal growth and promote legume growth. A wet winter will encourage more rapid cover crop decomposition, which will lead to earlier availability and higher availability early nitrogen. So we have to keep thinking about every year's conditions and putting it all together. So the C to N ratio and the tissue N content are good predictors of this plant available N. The younger the crops, the higher the N content of that species, the higher the availability following incorporation. And the legumes and mustards are going to have a higher tissue N content than cereals. The grasses will scavenge N, keeping N in the system, reducing that loss from leaching. And more biomass leads to more total N contribution. Not necessarily plant available, but just more total N contribution. And the soil moisture is going to be a driver for when and how much decomposition occurs. And whether it's incorporated or surface mulch will also influence when and how much decomposition occurs. I have a couple of resources, and I'll put these in the link in the chat. So this is just looking at doing some nitrogen calculations using your own biomass and tissue N content numbers. Here's this cover crop chart goes through, is created by the NRCS, and it's a guide to the characteristics and biomass and N expectations of many cover crops in California. And that's it. So thank you for your attention. And if we want to answer any questions, we might have a few minutes.
Yeah, we have a couple of questions for you. Thank you, Margaret. That was very thorough and clearly explained and put together a lot of details about nitrogen content of cover crops, very nicely laid out. So thank you for that. Here, we'll go with this first question. Well, you might have answered this one, but maybe you want to reiterate. Here's the question. I know you spoke to it, but are any other tips on estimating cover crop yields beyond book values? Do you ever cut quadrant squares for small areas and weigh biomass or do something similar?
Yeah, great. Well, I think I got to that at the end there. So yes, absolutely. I think just doing the sampling yourself, getting your hands dirty, paying $15 to get those numbers is really going to be extraordinary value. It is true that a lot of values, when you look them up on the internet, they could be for cover crops in any part of the country or even any part of California. And we all know how much the conditions influence the biomass and tissue content. So I do encourage you to do that sampling yourself. And like I said, I think it would take a small amount of time. You know, I've seen you can let your biomass sample basically just air dry, put it in a little protected place like a barn or a greenhouse or something, or in your kitchen table, let it air dry for a few days. I hope that answers your question.
Yeah. I like what you're saying. So you're kind of saying that people can kind of train their eye for estimating biomass. Is that kind of what you're saying? That's something.
Yeah, and I think what will be evident from this workshop is in the world of organics, we're doing a lot of guessing and estimating. And so there, the better we can get at refining our estimating and guessing, the closer we're going to get to understanding what's happening in our system. So, getting data from your own field is going to get closer to that number than using a table. But the tables are there for you regardless.
Yeah, all right, that's excellent. I know Eric Brennan told me about a method for drying biomass just using a microwave.
Oh, wow.
Maybe what we can do, we can send that method out. There's a paper, actually they use it for drying, estimating alfalfa. It came from that world. So we can send that out to people.
Okay.
After this.
Yeah, similarly, I've seen people use just the pilot light in their oven. Leave it open a little bit. Leave it cracked.
Yeah.
Right. That'd be neat to see. Thanks, Richard.
Safely, yes.
Yeah.
Okay, here's another question. Regarding the cover crop biomass nitrogen content table. Are those numbers collected from cover crop stands planted in rotation with vegetables following a vegetable crop harvest, or from a cover crop stand planted in a separate field, allowing them time to be planted on time and reach their nitrogen uptake potentials?
Yeah, so the tables that I have, that I showed are going to be estimates in a typical scenario on the coast or the Central Valley. So being more familiar with the Central Valley, I apologize. That scenario would be a fall planted cover crop. So November typically, just in time before our first November rains. And then typically they're harvested in late March, early April. So that would be the classic scenario, and that's what those numbers would represent.
Here's a question that I know you know something about, Margaret. Can some of the legume cover crops have influence on soil disease?
Sure. So that was the subject of some of my PhD work, looking at the potential for legume cover crops to support the growth and development of Verticillium dahliae, the causal organism of Verticillium wilt. So I did find that common vetch in particular was a good host for Verticillium dahliae, and that it did not show symptoms. So if there are pathogens of concern, then that would be worth asking and exploring, asking your local advisors or forms of support or searching that online. There's not, I couldn't generalize. I think it really comes down to what pathogen you're concerned with and understanding what crops are a potential host for that. So I think that's an important nuance to really know what you're going after. And similarly, it doesn't stop with soil-borne organisms or pathogens, but insects. And so in my area, for example, we have a lot of issues with tomato spotted wilt disease, with tomato spotted wilt virus, and fava beans are a host for that. And so folks often will avoid fava beans in their mixes because of concerns over tomato spotted wilt virus. So there are local concerns and issues specific to your system. So that is important to keep in mind. And sorry, one more, we tend to avoid some of the brassicas up here because of concerns over brigada bugs still and other types of insects, even aphids coming in early on fava beans. So yes, it's important to keep all the insects and pathogens in mind when selecting your cover crop.
That question comes up here in particular, Margaret, because of the strawberries and verticillium, you know, so that's a huge concern, I think, that's on people's minds. And so that's kind of steered us away from legumes in cover cropping in this area. So, okay, here's another one. What determines the ideal C to N microbial diet? I have seen other values like 11-to-1 instead of 24-to-1 at the FREP conference.
Yeah. Well, I think Louise will probably get into that. So I'm going to defer that conversation to Louise's next segment. So she's going to go deep into the microbes.
Okay. I see that Eric just put on the chat the microwave drying method. So that's there. So thank you, Eric, for doing that, Eric Brennan. And let's see, what time is it? How about this next one? Maybe Margaret, if you wouldn't mind just answering it on the Q&A. It's because it's time now for us to move on to Louise. So thank you very much, Margaret. That was excellent.
Great. Thanks, everybody. I wish I could see you, but nice to be here with you.
Yep. Okay. Well, now we will move on to Louise Jackson, and she's going to talk to us about the role of soil microbes in providing plant available nitrogen. So, Louise.
Hi, everyone. Well, it's really nice to be here, even though it's virtual. I want to thank Richard and Joji for setting this up and for inviting me to be part of this. I am now retired, but many years ago, I did work in the Salinas Valley, and I'll talk a little bit about some of that specific work. So my title is The Role of Soil Microbes in Providing Plant Available Nitrogen. Okay, so why isn't it? Okay, here's my outline. I'm going to be talking about these topics. First, about root uptake of soil nitrogen in vegetables, then a close-up look at roots and soil microbes in the soil, how and why soil microbes produce plant available nitrogen, and then ways that the microbial symbiosis has improved nitrogen cycling. And really, I'm just adding on to what you've heard in these excellent talks by Daniel and Margaret. And I just hope to embellish it a little bit. There's so much overlap. So in the picture here, we see a microbiologist view of a plant and a growing root, microbes around that root, and then looking at a smaller scale at the arbuscular mycorrhizae that can grow there. And what this diagram doesn't show is the messiness of soil. So I'm going to try to put that together in my talk today. So the first topic is root uptake of soil nitrogen and use of nitrogen in the plant. So as you've heard before, ammonium and nitrate are the end forms taken up by roots of annual plants. And ammonium, NH4+, is positively charged, nitrate is negatively charged. And the compounds also vary in a really important way in that ammonium is more reduced, essentially has more energy in it than nitrate, which is more oxidized. Ammonium in some ways seems like a great bet for a plant since it's already reduced, but it can be toxic. And under high ammonium conditions, we want to avoid toxicity. And the general rule is that plants taking up a mix of ammonium and nitrate is better for maintaining pH within the plant. In the annual plants that we're working with, we just don't see much in the way of organic forms of nitrogen. In other words, compounds that contain both nitrogen and carbon. It's these mineral or inorganic forms that we see. So this rather daunting diagram on the right shows the pathway of soil nitrogen uptake by the root to plant proteins. And the important message I want to give is this is a very energy consuming process. In fact, some people say it's one of the most energy intensive processes in biology. So when plants take up nitrate, they use a lot of energy and enzymatic reactions to form ammonium. And that ammonium then can be bound into amino acids, such as these, which then are formed in polymers into proteins. What's key here for the plant is the photosynthetic energy. The photosynthate produces the energy that can cause this reduction process. And it also provides the backbones of these precursors to amino acids. So the main point here is that this is the way that the plant builds its organic matter, largely based on the energy from photosynthesis. So now what we're going to do is look at the root in soil. So not just the rhizoplane and the root hairs that we just talked about, but also the microbial community and soil food web around the root, and why it is that plants, especially in organic systems, get a lot of their nitrogen from microbial processes. So this diagram, it's not to scale, but it's really informative in thinking about the plant microbial and soil food web little microcosm. The tan pieces are sand and silt particles. What we see are clays built up with organic matter. It's a clay organic matter complex in the more gray colored material. That adds a lot of structure to the soil. And in between these mineral particles, we see pores that are either, in this case, open, suggesting they're more aerobic, or with water films around them that are important for the life in the soil. So in fact, maybe this is the situation that might be best for both microbes and plants, with aeration and water films for their metabolism. The plant root is shown here. You can see root hairs, bacterial colonies along here. Mycorrhizal hyphae grow into the root, and in this, we'll talk about this symbiosis a little bit later. Microbial colonies are also found, say, around decomposing plant cells in the nooks and crannies of this clay and organic matter complex. Fungal hyphae and spores are around this same organic matter complex in the decomposing cells. Fungi bind this together. But what's really important in this whole cycle is the role of predators. So nematodes are organisms that can consume bacteria and fungi. They can also consume other nematodes. There are protozoa such as amoeba and ciliates that engulf bacteria. And then there's microarthropods that can consume all of these different forms of life. And the biodiversity in soil aggregate is immense. Not only are we just beginning to understand it, we're beginning to understand just how crucial the environment is, as wet-dry cycles can change the community composition of the microbes. And I'm not going to have time to talk about that, but what I will talk about more is the soil food chain. So if we look at a soil organic matter molecule, and here's just a hypothetical one here, shown here with this little star shape. Soil microbes such as bacteria and fungi can break down that organic matter, as we've heard about, to meet their own carbon and nitrogen needs. Then organisms on the right consume the organisms on the left, and this process goes up through several tiers, trophic levels. So here are organisms that consume the bacteria and fungi. We see omnivores that then consume some of these organisms, including the amoeba, different types of nematodes. And finally, on the far right, predator organisms, which actually consume the omnivores and larger creatures. The arrows are really important. And what I just described is carbon and nitrogen going from the microbes to the higher trophic levels. But when these organisms are consuming their prey, there are waste materials, for example, that contribute back to the organic matter. So this organic matter is in continual flux, not just in terms of the amount that's moving through the food web, but the types of organisms that are being decomposed and their waste materials that are being contributed. So what's important here is the idea that this is a very dynamic situation. We often talk about it as more stable than it really is. Changes can occur just within a few hours. This is quite similar to some of Daniel's talk, but what I want to mention is the types of organic matter that are really important in the nitrogen cycle. Soil organic matter is composed of carbon, nitrogen, oxygen, hydrogen for the most part. And most of it is what we might consider recalcitrant and protected soil organic matter. Things like humic substances that are really hard for microbes to break down. In fact, most of the organic matter is in this state. And it's super important for agriculture in that it has a lot of importance for structure, for aeration, for infiltration of water, those clay organic matter complexes that was in the diagram. But much less abundant is what we'd call active soil organic matter, sugars, amino acids, very readily decomposable plant material, dead and live microbial cells. And the importance here of emphasizing this is that this is mainly what gets into the nitrogen cycle. So what microbes do, as Daniel explains, is break down the soil organic matter to get soluble or available carbon for their growth and maintenance. And nitrogen is also released. That nitrogen can be taken up by microbes to meet their nutritional demands, or it can be available for plants and other microbial processes. So this nitrogen cycle shows the soil organic matter is the kind of anchor point. Mineralization you've already heard about from Daniel. It's the process whereby this organic nitrogen is converted to mineral forms, ammonium, as microbes are breaking down this material to meet a myriad of different needs. That ammonium can be bound to the cation exchange capacity, which is largely negatively charged soil particles in the soil. It can be volatilized, but at the pHs that we just heard about, volatilization is really not an issue. Ammonium, it can be thought of as an energy-rich compound, can also be nitrified by microbes. Nitrification is the process by which ammonium is converted to nitrate. The ammonium and nitrate that are formed during mineralization and nitrification obviously can be taken up by plants. A mix is great for the pH balance. Also can be taken up by microbes to meet their nitrogen demands. But there's an important distinction, and that's that microbes will tend to prefer ammonium. Why? Well, it's more energy rich. Microbes don't have the luxury of the photosynthate that plants do. Their energy is coming from the carbon that they're breaking down. So there's a tendency for microbes to prefer ammonium as the nitrogen source. If nitrate builds up in the soil, we can see leaching and denitrification if the soil becomes waterlogged. And that's what we want to avoid, not just because of the environmental consequences, but because, especially in organic systems, this nitrogen is very prized and we want to keep it there. Denitrification is the process where under anaerobic conditions, nitrate serves as an electron acceptor in metabolism, sort of like oxygen would in an aerobic condition. Now let's look a little bit at these arrows. As plants and microbes die and turn over, they return to the soil organic matter. And it's important to realize that these communities of microbes are turning over really fast. And unlike plants, there's not a pool that necessarily accumulates in proportion to the rate at which they're mineralizing in. just some quick definitions that you're probably really familiar with by now. Mineralization is the breakdown of soil organic matter that relates in mineral ammonium release. Nitrogen immobilization is the microbial uptake of nitrogen to meet the nutritional needs. Nitrification is when microbes rely upon ammonium as an energy source that's oxidized to nitrate, and denitrification occurs under waterlogged or anaerobic conditions when nitrate is reduced to N2 gas, which is not an environmental issue, but if it's nitrous oxide, that's a greenhouse gas, and that is a pretty potent greenhouse gas that we'd like to avoid. So I'm now going to tell you something about a study that you just heard about from Margaret, and I'm just going to give you more detail on it. And I'm very glad that she gave you that introduction. And here we go on this cover crop that's grown on Chular loamy sand. This is a very old study. I apologize for it, but I don't think anybody else has done something like this since. So what we see here are Phacelia and Merced rye cover crops, and this center strip in here did not have a cover crop. What we're doing is we're pushing in cylinders so that we can apply an isotopically labeled cover crop, very safe, heavy isotope of nitrogen. And then look where that went in a contained space. And here's a picture of John Inman, bless his soul, that was kind enough to push in these 2-foot cylinders into the soil. So roots of and shoots of Phacelia, which is a cover crop that was probably about 3%, and it's not a legume, but it's very fleshy and easily decomposable. It had a C to N ratio of 19, was incorporated into the top of the soil cylinders. And then iceberg lettuce was seeded 45 days later. Here's the inputs of fertilizer and water that were given. Second iceberg lettuce crop was then grown. Here's some of the methods. And I need to mention that at the base of the cylinders, we put in these little resin bags that are able to trap nitrate. And so we could look at the amount of the cover crop that went into the, that was leached. Well, you just saw this slide, so I will go over it again. It's always good to get some repeat on these talks. This is the amount of nitrogen 15 from the Phacelia that was in the first lettuce crop. So 60% was still in the soil organic and pool, 20% went into the lettuce, very little in the inorganic and or microbial biomass. About 5% was leached to 2-foot depth, and about 11% was unexplained. That unexplained could be experimental error. Maybe we just didn't get all our N15 back as we excavated all the soil out of the cylinders, but it also could be partly to denitrification that occurred during the times when the cylinders were being irrigated. So one interesting question is, why is there so little inorganic and microbial biomass in the soil when we know that there was a lot of mineralization that went on? Otherwise, we wouldn't have only 60% of the N15 left in the soil organic matter. And this just shows the fact that the nitrate in the resin bag at the base of the cylinders was quite low in Phacelia and 15. There was some nitrate leaching that occurred, and that was probably from the soil that had been in the cylinders prior to the onset of the experiment. It hadn't had a cover crop to extract that ammonium and nitrate. Here's a picture of the resin bag that was responsible for this data. So now let's go back to the nitrogen cycle that we just showed and said, we have Phacelia here, not exactly soil organic matter, but on its way to becoming soil organic matter. So 60% of the N from the Phacelia is still in this organic matter pool. Mineralized N occurred on 40% of the Phacelia. It was mineralized pretty rapidly in order for plants to take up 20% of it, but not much is accumulating in the microbes themselves. Why? Well, a lot has to do with the fact that the plants are accumulating over a period of weeks. Microbes are turning over really rapidly. They're turning over because they've been preyed on in the soil food web. They're preyed on because they might be abundant in certain locations where they can be more accessed by the members of the soil food web. Also, things like wet-dry cycles, flooding, the disturbance of soil by weeding can perturb the microbes, and they'd start to turn over, and other members of the microbial community can replace them. So what we're seeing is a situation where the mineralization and nitrification process is going on pretty quickly. It's going into the microbes, but there's also a very rapid turnover, so that the accumulation of N15 at any one time isn't anything like what the Phacelia picked up. And the result of that, with the lettuce being very active with its roots in the right place at the right time, was that there was relatively little loss of the Phacelia cover crop N by leaching and denitrification. So what are the management implications? One is that plant soil microbes require easily degradable organic matter to supply crop plants with N. And I've just made a long list of the different kinds of materials that are important for that active soil organic matter to be available to nitrogen for mineralization. I think it's really important, even though it's quite hard to measure a lot of these things, to think through when these different soil organic matter pools might be more prevalent in your cropping system and manage accordingly. When is it going to be really likely to have rapid mineralization? We've heard some of the examples already. And when is it going to be important to tie up some nitrogen so that the microbes are not moving into a really active mineralization mode? One thing that we found in this and other studies using N15 is that low concentrations of soil ammonium and nitrate can be okay in organic systems if organic matter mineralization is rapid and plant uptake is rapid at the same time. What is happening is there's just continuous flow through the nitrogen cycle, and those pools don't build up. But what you have to make sure of is the crop is getting adequate levels of total N. Some organic systems, especially in their inception, have low concentrations of soil ammonium and nitrate because there's just not the N supplying power from that active soil organic matter fraction yet. One thing we've also found too is that petiole nitrate is not always a dependable assay in organic vegetable production. It's possibly because plants are using more ammonium. So one of the messages that I think you've already heard is to minimize the nitrate losses from these systems is that it's important to really manage your crop residues for the different phases of the farm's cropping system and different seasons of the year. Now I'm going to talk about ways that microbial symbioses improve nitrogen cycling. So here's a diagram of the arbuscular mycorrhizal fungi and a little more detail here. So for arbuscular mycorrhizal fungi, which are very specific type of species, the spore germinates on the outside of the root, and then hyphae grow like here in between the cell walls. Once they invade a root, a structure is formed called an arbuscule, in which the plant begins to supply carbon to the fungus, and it grows out into a much larger soil volume than would be accessible by the root. And in return, brings back compounds, nutrients such as nitrogen, phosphorus, and zinc. And lettuce can become very highly colonized to its benefit by mycorrhizae. We found that it can increase yield in some situations in tomatoes, but more important, it's more that the crops are more nutritious, and there's a tendency for there to be higher water use efficiency and a decrease in nitrous oxide emissions with AM fungi colonization. So what can we do to increase the benefits from it? having more crops in the rotation with the ability to form the symbiosis. Canopods like chard and spinach do not have the symbiosis, nor do brassicas. So a more continuous cropping system with these hosts means that there's like fragments of roots and spores that can colonize the next crop. It takes time for these fungi to colonize. We've saw in a Tanner and Antle set of fields that there was very little at the beginning of conventional organic transition, but then after 3 years, there was 10%, and it can get higher than that. It can happen without inoculating the soil. Lots of information shows that continuous cropping with less tillage and less fallow is conducive to the symbiosis. Just briefly mention nitrogen fixation by legumes. Over here on the left, what we see are bacteria invading the root through root hairs. They put infection threads into the root and then colonize inside cells to form the nodules that Margaret also showed you. And inside that nodule, ammonium is produced from N2 gas. And it's no surprise after what I've emphasized so far that, and Margaret's already discussed, is a very high energy demand process. And the result is a source of nitrogen to the legume plant that wouldn't otherwise be there and to the soil as its residues decompose. Margaret's already emphasized the idea that legume cover crops in mixtures with higher CDN species can be good for reducing the really rapid mineralization of these species in the soil. And it's good to avoid a long lag between the legume cover crop and the real crop to minimize nitrogen loss from premature mineralization before the roots are there. And transplants might be better than direct seeded crops at obtaining legume-derived N because their root system is from the get-go more advanced in the soil and exploring it for nitrogen. Well, that's the end of my talk. I want to just step back and say a couple of conclusions. One is the overall goal of all of this, I think, is to provide adequate crop intake and growth in organic crops with high-end mineralization of soil organic matter occurring at the time of peak crop nitrogen demand. And that then has to be matched to the time when the plant can reach its maximum uptake potential of ammonium and nitrate. And in turn, that can result ultimately in lower losses of soil nitrate and keeping the nitrogen in the system for as long as possible. So to increase the role of soil microbes in providing plant available N, what's crucial is active soil organic pools and a dynamic soil food web that's keeping the system moving through these various phases. And that can take time in an organic system, especially the creation of a more complex soil food web with organisms as complex as predators. We can also increase this with the microbial symbioses that I just talked about. And something that I haven't emphasized enough is the role of environmental conditions within those soil aggregates, including soil texture, how much clay there is, how much that absorbs ammonium, and how clays form organic matter complexes. How compact is the soil, how aerated it is, what kinds of water infiltration there is. But I'm going to stop here and answer any questions you may have. Thank you.
Thank you, Louise. That was very interesting. That's, you know, kind of the thing that is the hardest thing to understand in this whole nitrogen cycle is it's just so dynamic, and the role of the microbes is so, you know, it's at the core of the whole thing. And so we appreciate you sharing your knowledge on that. And let's see if we have any questions here. I have a question. What is the, you know, like if you're if you're trying to understand how you're doing with your nitrogen with the microbes in the soil, in your soil management, what do you look at as indicators, good indicators for understanding, like let's say you wanted over a period of years to be monitoring the health of your soil microbial populations in the soil?
Well, this is something that Daniel already mentioned, is the fact that we don't really have a lot of soil testing going on for these kinds of pools, the really labile pools that are really what drives a lot of the microbial nitrogen cycle. I think we want to see changes in soil carbon, but often that doesn't occur for the first maybe even 3 years. So you can be doing a lot to your soil and adding these pools of soil active material, but it's not showing up in your testing. So I can almost say one tip is don't be disheartened if your soil carbon doesn't show up really quickly. It's going to take a while.
Mhm.
Another thing that can happen is that you can start noticing by paying really close attention to the nitrogen content in the leaves. Like you can take samples more frequently during the season. Sometimes in organic systems, plants are a little bit slower to take off. Sometimes I wonder, is that partly part of the mycorrhizal symbiosis kicking in? But it's often hard to quantify what's really going on in these pools without doing the kind of research that we're talking about here in this talk.
Mhm. Yeah, this comes up a lot with growers, just what's a good way to measure the health of your soil. I mean, this is something the NRCS has whole, procedure for doing and measuring soil health. And
Yeah.
it's a big area of kind of people are trying to understand it. And I would hope in the future that, like some of these tests, like Daniel talked about, that FDA test becomes more available. Here's a couple of questions. What role does compost or similar amendments, such as biochar, play in supporting these microbial processes and nitrogen cycling?
Okay, let's just talk about compost first. A lot of the compost that you add is already pretty, there's a lot of mineralization that's already occurred, and it's pretty stable. But there are compost compounds that are more soluble and more readily decomposed and mineralized by microbes, even when you put it freshly in the soil. I think one of the key things with compost is to realize that you're not going to see the instantaneous reaction that you would with some of the more soluble plant based or other materials that a lot of organic farmers have to use, especially initially, to boost the N supply to the plants. The second question was about biochar. I haven't worked a lot on biochar. I do know that biochar can differ a whole lot between different sources and different manufacturing processes. It does form, like the cation exchange capacity, a possibility for ammonium adsorption. And it also apparently provides microsites for microbes to hang out. So I think that it's something where if you have access to it, that you can try it out slowly on your fields and wait again for a year or two before you make a decision that it is really effective or not.
Okay. Here's a question. What % nitrate to ammonium do plants tend to utilize?
Oh, that's a good question. That is one of the hardest things to measure. It's really, really hard to measure because think how dynamic it is. You put nitrate or ammonium into the soil and it's immediately going off in all these different directions.
Mm.
But what we do know from solution culture in lettuce that it's good to have a mix. And a lot depends on whether or not it's really soon, say after a cover crop is added or later in the crop cycle. It could be that with time, more and more nitrate dominates the system. The reality is that in most conventional systems, plants are relying on nitrate. Why? Because that ammonium that's being produced in mineralization is just so important for nitrifiers. It's a very high energy compound, and plants may not get a crack at it before the microbes are already converting it to nitrate. There's some estimates that if a plant relies totally on nitrate, it can use like 20% of the plant's energy to use nitrate alone.
So some ammonium is beneficial. Yeah. Okay. Very good. You got a few here, Louise. What is the best way to improve diversity of soil microbial food web, and how best to measure diversity?
Some of the work that we've done on processing tomatoes suggests that the more diverse the inputs you add, the more likely it is to have greater diversity. So in other words, using legumes, using other types of cover crops, having different kinds of organic matter inputs, whether it's poultry manure or other kinds of manure at different times of the year, may increase diversity. However, measuring diversity is really hard. We can use markers such as phospholipid fatty acids for changes in the whole microbial community. Doing the actual DNA work is really hard, mostly because we don't even know a lot of the information that we need to about the microbes. So there is a major obstacle that we've observed in organic farms, and that's that it often is hard to reach the stage where there's a lot of predators. And the predators in the soil food web are really important for regulating the different populations that are there. So that is a job for somebody down the road, is what can we do? Do they need to be introduced? It's like, what can we do to further promote the diversity in the soil food web?
Well, that kind of ties in with this next question, because he's asking, there are many microbial products that claim to improve the microbial ecology of the soil. Have you ever found that these types of products have a beneficial effect?
Well, that's a really hard question. It's, there's that adage of feed the soil. And I think that for these kinds of products, and there's so many of them, I'm not going to put them all in one basket. For them to be effective is they need active soil organic matter. And the relative efficacy can have a lot to do with what environment they're put in, as well as exactly who they are. Mycorrhizal fungal inoculum does inoculate plants, for example. But as far as some of the mixes and who survives, that's really hard to say.
Okay. Here's one. Plant tissues in organic systems tend to be somewhat more robust, tougher, and tougher compared to the same varieties grown conventionally. What do you attribute that to?
Oh my gosh. Well, this is really a hard guess, but I think that if the soil organic matter is supplying a lot of the nitrogen and other nutrients, that there can be higher nutrition inside in organic vegetable crops after the system's been stabilized for a while. The first onset of organic production is probably not going to contribute that same set of traits to the plant.
Okay. And it looks like we have time for just this last question, actually, so this works out well. And he says, thank you for your pioneering research on cover crops. What do you think is needed to increase the adoption of cover cropping in major vegetable production areas like the Salinas Valley?
I'm not sure how to do it, but I think we need to figure out how to make it even easier than it is today, and make it worth people's while to put in a short-term cover crop, as Margaret talked about, one that's still at the stage of accumulating nitrogen and is able to be incorporated on top of beds in an easier fashion than just disking up the whole field. So I think that it's not that people don't think about it, it's that it can really interfere with what they have to do.
Yeah, I like that idea of kind of not no-till, but some kind of a minimum till way, like the work that you did with incorporating the Phacelia on winter beds. I thought that really opened the door for people to look at ideas for getting cover crops into our rotations here. Yeah. Well,
Yeah, it's still not that much, so.
Well, thank you very much, Louise, and thank you to all of our speakers today, to Louise and to Daniel and to Margaret. And Rachel, I think we'll turn it back over to you, and we need to see that code. People need to be able to scan that. And so thank you all for attending, and we will continue this series next Tuesday, same time. And we're going to have Patricia Lazicki talk about some really nice research that she did on the mineralization and nitrogen release from organic amendments. Margaret will talk about nitrogen release from compost. And then Mike Kahn will give a roundup about nitrogen from the amount of nitrogen in irrigation water and its effect on plant growth. So any final comments, Rachel?
We'll see you all next week.
All right. Thank you very much, everybody.